Okay. Good evening. Can you hear me? Okay. Today we have very simple lesson. Today morning lesson very important. I hope you all completed up to secondary growth and root structure. Right. Up to root structure we complete. Root structure we complete. That means I have sent all diagrams. Plant leaf structure, stem structure. Root structure and stem secondary growth I have seen it go. One of this diagram definitely come in the exam. Okay, let's move into forest. Okay, the forest part very simple. We have only three main types of forest. Okay, now more than 80% of this lesson we have completed. Okay, up to this level we discussed. Okay, forest. Okay, actually for the economical balance and environment, to keep the environment balance and keep biodiversity and protect very valuable genes. To protect valuable genes, give me a second. Okay, sorry. Okay, now I told you to keep the environment and environment balance and ecological balance and to keep the biodiversity and to protect valuable genomes. Very important this forest in Sri Lanka, not only Sri Lanka, entire world. This forest very important. The forest, the definition is given: a population of plants belongs to the different regions can be called as forest. This forest mainly we can categorize into two groups. The first one is primary forest and the second one secondary forest. Secondary forest. Primary forest mean, it's simple. Primary forest mean, what's the meaning of primary forest? That means no human invention. It's completely natural forest. Completely natural forest we call as primary forest. After, due to any reason, forest affected by man, after human invention and growing a forest called as secondary forest. Secondary forest. Cultivation, obtaining woods or food supply. They are secondary forest. Naturally, as example, Sinharaj is a primary forest. Primary forest. If there are any cultivation to get food, wood or food supply, they are called as secondary forest. Okay, they are the main difference between primary forest and secondary forest. If it is primary forest, no human invention, no human invention. But secondary forest has been influenced by man, right? It is made by man, right? It is completely natural. In primary forest, the biodiversity is great, different types of plant varieties, different types of valuable animal types, high biodiversity. But biodiversity is low in secondary forest. Secondary forest means same type, same variety plantation, right? Same variety plantation for get wood, mainly for wood, right? To get wood. There are different types of plants in primary forest, but in secondary forest, same type of plants, and plants heights and ages also difference in for primary forest. Secondary forest mean it's same age. Now then, did you collect it? Then others you all got right. Yes. Send message. Okay. Secondary forest, all plants in same age and same height. No variations. Therefore, biodiversity very low. Biodiversity very low. Okay. And the competition for nutritions also still didn't receive. It's a problem. Wait. Say first 1, 2, 3, 4, 5, 6. Okay, only you don't. Did you check in the post office? Okay, I think post office problem. Everyone got others. Hari, Palam, you have soft copy, right? Use soft copy and make a short note. Actually, it is very long tute. It's a bulk tute. Make a short note. You have soft copy. Hari. Okay, they are the differences, and these plants are if it is primary forest, different age and different height plants. That's why its biodiversity is great. But secondary forest, same height plants and same age, same characteristic. Therefore, what happened? New competition for nutrition, high competition for nutrition. The secondary forest, but primary forest, different root zones. Roots, sometimes this fibrous root system, sometimes very deep taproot system, sometimes shallow taproot systems, right? Therefore, no competition, no more competition, right? But in secondary forest, plant roots are same layer, growing on same layer, same deep, same depth. Therefore, highly competition for fertilizer. Therefore, we have to add some fertilizer for secondary forest, but primary forest naturally circulated. Naturally circulated. Okay, they are the differences between primary forest and secondary forest. Very simple. Okay, let's see what are the difference. What are the main types of forest in Sri Lanka? In Sri Lanka, mainly three types of forest. Mainly three types of forest. The first one, tropical wet forest, tropical wet green evergreen forest, tropical wet evergreen forest, tropical wet evergreen forest, and dry mixed evergreen forest, and hill country wet zone forest. Okay, what are the main three types of forest? Tropical wet evergreen forest like Sinharaja Khannelia, dry mixed evergreen forest, and hill country wet zone forest. Hill country wet zone forest. Hill country wet zone forest. Okay, let's see what are the characteristics and their plant types and what are the examples for them. That's enough, right? Very simple part. Okay, let's see what are the tropical wet forest, tropical west evergreen. Their main characteristics are tree heights between 30 to 40 meters, and there are several layers. We can see layers clearly like this. Yeah, they are the layers. They are floor layer and understory layer, canopy layer, and emergent layer. Yeah, we can clearly see layer differences. Clearly see layer differences. See different layers. There are several layers. Yeah, mainly four main layers and five sub layers. Okay, clearly we can see layers in this tropical wet evergreen forest like Sinharaja Khannelia, Dediyagala, Nakiyadeniya, Morapitiya, Bambarabotua. Okay, they are the examples for tropical evergreen forest. Tropical wet evergreen forest. Sinharaja and Khannelia are the main tropical evergreen wet forest of Sri Lanka, right? Additionally, Dediyagala, Nakiyadeniya, Morapitiya, Bambarabotua. And main plants are Hora, Keen, Na, Godapara, Mill, Midel, Kitul, and Nadu. Okay, they are the main plants. Okay, they are the characteristic of tropical wet evergreen forest. It's clearly we can see layers. Foot layer, they are the layers, right? Foot layer, understory layer, canopy layer, and emergent layer, right? They have huge competition to get sunlight. That's why the plants grow layer by layers. And color of tree are dark green. Yes, the color of trees are dark green like this. There is a canopy emerge at the top of the no light inside the forest. Yeah, little bit dark color in inside the forest because the light transformation very difficult due to several layers. Plants growth very dense. Yeah, then you can understand like this. And higher biodiversity, yes, comparatively high biodiversity, very rich in biodiversity, rich in biodiversity. Rich in biodiversity. Okay, I have missed one point. Please note down here. This tropical wet evergreen plants stems, plant stems, plant stem same diameter, same diameter. Same diameter. As example, if you got horror tree, horror plant එකක් ගත්තොත් it's same diameter. ගොඩාක් උසට යනකම් same diameter. Right? That one also one characteristic. That one also characteristic, no doubt. Okay, another characteristic is the edge of plant leaf is well sharp. Well sharp. Again, the edge. See, this is the edge of plant leaf. The edge of plant leaf is very sharp. Well, sharp mean like this. This is the shape of plant leaf. This is the shape of plant leaf. This is the edge. This is the edge. This edge is sharp end. Why? In wet zone, normally this forest are heavy rainfall. Heavy rainfall. Every day raining. Every day raining. Therefore, the extra water is accumulating on the layers. On water drops are accumulating leaf blade, leaf surface, right? To remove this excess water, the edge of plant leaf is sharp to remove extra water accumulation, right? Again, වතුර එකතු වෙන එක නතර කරන්න. Right? If it is accumulate water, it's burning with sunlight. This leaves are burning due to focusing sunlight into leaf. Right. Hi. That's about tropical wet evergreen forest. Tropical wet evergreen forest. Let's move into dry mixed evergreen forest. Dry mixed evergreen forest. Dry mixed evergreen forest example Somavati, Vilpattu, Maduroya, Udawalawa, Vasgamuwa, and Mahavali areas and Yala. You can remember these famous things. Somavati, Vilpattu, Udawalawa, Yala. Right? They are dry mixed evergreen plants. It contain different plants, satinwood, ebony, veera, suryamara, milla, palu, like famous plants, right? Famous plants. And what are the features? What are the features of this dry mixed evergreen forest? Okay, the difference is the forest light green color, not dark green, not dark color inside the forest. And there are hardwood tree. and they don't grow dense, and they are tall trees at intervals with gaps. Tall trees. Therefore, sunlight directly coming inside to the forest. Therefore, no darkness. No darkness. The other thing, they are not same diameter. The plant stem not same diameter. You can note down this. Plant stem not same diameter. Like this. The shape is not like this. Shape is, this is a shape. This is a plant stem shape. Plant stem not same diameter. But in wet evergreen forest, it's same diameter. Plant stem is not same diameter, it's changing. It's changing. Okay, they are the difference of dry mixed evergreen forest. Okay, let's see what are the examples for hill country wet zone forest. Okay, you already know Samanalakanda means Shripad area, Adams Peak area, and Pidurutaragal. Yeah, it's a hill country. Knuckles and Horton Place. Easy to remember, right? All mountains. All mountain forest are wet zone, hill country wet zone. Okay, what are the plants? Valsapu, Keen, Mihiriya, Devata, Berelia, Mora, Hulang, Kiria. Sorry, Hulang, Keek mean. Okay, what are the features? The most important feature is they have a continuous canopy. They have continuous canopy like this. Completely one layer, as a one layer, continuous canopy. Continuous canopy. Colorful leaves can be seen. And remember, if you got plant stem is due to the high wind, normally hill country wet zone, they have very speed wind paths. Therefore, their stems little bit what we call that no straight stems මෙහෙම no straight stems like this. See, stems නිකන් ඇඹරිලා ගිහින් තියෙන due to high wind. No straight stems. The stem like this. See, no straight stems. Okay, that's the shape of stems of plants. Okay, and they have very small leaves. Small plant leaf. Why? Small plant leaf for retained for the high wind conditions, high speed wind conditions. To tolerate high wind condition, they have small leaves, and they maintain a continuous canopy, right? Continuous canopy. Okay, importance of forest. You already know. It's maintain oxygen carbon dioxide balance, and it's protect soil erosion and maintain soil fertility, and it is increase biodiversity and protect valuable genomes, and protecting water, water resources, and provide valuable woods, and provide protection for pollinators and seed germinators, and for tourism industry, for natural beauty, and protection of endemic species. Mean අපි කියන්නේ වඳ වීගෙන යන අය කියලා. Yes. To protect these genes, biodiversity, very important this forest. Okay, that's a forest lesson. Very simple forest lesson. right? You have to remember some characteristic of each forest and example. Examples තුන හතරක් මත තිබ්බා මෙතන. Right? These plants නම් අහන්නේ. හරි? Okay, let's move into last two lesson, tissue culture. Okay. Tissue culture. Okay, tissue culture. Always asking this question. Tissue culture, always asking tissue culture questions. Balum, what's the meaning of tissue culture and their applications? Okay, tissue culture mean it's a technique of plant propagation. It's an artificial plant propagation technique. Plant tissue growing in an artificial media. Plant tissue part, very small leaf part or root part or stem part or whatever plant tissue growing in artificial plant media inside bottle in artificial conditions. Okay. And aseptic condition, we have to make aseptic condition. Aseptic condition mean without microbial attraction. Tissue culture refers to technique of growing cells, tissues or organs on a nutrition medium, artificial nutrition medium under sterile conditions. Sterile condition mean without microbial infections. This method we called as micropropagation. Yes, micropropagation mean very small cell grow as a complete plant. This ability we called as totipotency. Okay, what's the meaning of totipotency? Totipotency mean one single plant ability to develop as complete plant. ඒ කියන්නේ එක cell එකකට plant එකක් විදියට grow වෙන්න information එකක් තියෙනවා. Genetic material, all ribosomes, everything is there. One cell able to develop as complete plant. This characteristic, this property we called as totipotency, the ability of single cells to regenerate as whole plant, that we call as totipotency. This totipotency characteristic of plant cell utilized for this tissue culture process. Tissue develop as whole plant, tissue part of plant develop as whole plant. Okay, this tissue culture very simple. What we doing? The first thing we have to separate explant. How to select explant? Explant mean explant, right? Explant කියන්නේ the explant is a plant part, mother plant part. Select for grow in the artificial media, right? It's a very simple thing. Explant is a small piece of plant tissue or cells taken from the mother plant for the start the culturing process. In tissue culture, callus is soft. Callus I will explain later. White mass of plant cells that grow when the piece of plant and keep nutrition medium undergone sterile condition. Yes, sterile condition mean without microbial infections. බලන්න. Okay. They are the main steps of tissue culture. If you got this story, that part also very simple. In tissue culture, what happened? We get plant part, tissue part from mother plant and cultivate in artificial media and sterile condition, and we keep in artificial environment, control environment එකක තියලා ඒක grow කරනවා inside a bottle or a test tube, right? That's the meaning of tissue culture. To carry out tissue culture successfully, the following is required. The first one, explant. What's the meaning of explant? Now you already know. Explant means the plant part separate from the mother plant. the plant part separate from the mother plant. The second one, the nutrition medium. The nutrition medium mean it's an artificial media that we call as agar media. Agar media එකක් කියන්නේ semi-solid media. It's contain all plant nutrients, vitamins, hormones, everything, right? Everything required for plant growth available in tissue culture artificial media. Okay, hari. මෙයත් හරි. මම පස්සේ එකක් කියලා දෙන්න. And growth regulators. Growth regulators mean hormones. Hormones are mainly three types: auxin, cytokinin, and ghrelin. Auxin, cytokinin, and ghrelin are the. I will explain one by one what is the purpose of them. Later, now understand that's one also requirement, right? And carbon source. The carbon source also there. The amino acid, vitamins මේ ඔක්කොම තියෙන්නේ මේ nutrition medium එකේ, right? Solidification agent විදියට අපි ගන්න agar. Proper physical environment. Proper physical environment mean we have to maintain light, pH value, oxygen level, everything artificially controlled. Strict aseptic conditions under laminar flow hood, laminar flow cabinet. Within bracket cabinet. I'll show you everything. Laminar flow cabinet. Normally, every tissue culture process, tissue culture explant preparation, media preparation, and inoculation. Inoculation means this explant deposit on the plant, growing media. Everything we doing subculturing. I will explain now. Everything we do in this laminar flow cabinet. Laminar flow cabinet is just electric cooling. It's a box cabinet. What happened? This suck all air inside it has a mechanism suck all air and provide sterile air into it's just like air circulation process, air filtering process එකක් තියෙනවා ඒක. එතකොට ඒක ඇතුළේ තමයි අපි මේ ඔක්කොම කරන්නේ. Then we can maintain aseptic condition. Right. Okay, let's see one by one steps of tissue culture. The first one, select the tissue culture explant. Okay, what is explant? Explant විදියට ගන්න පුළුවන් අපිට. We can get explant as seed. මෙන්න මේව ගන්න පුළුවන්. Leaf piece, shoot tip, root tip, young stem, inflorescence, flower bud. මේ ඔක්කොම නෙමෙයි, any one tissue or seed, embryo, fruit pot, pollen grain, any cell of plant we can collect as an explant. The main thing, this explant should sterile conditions. What's the meaning of sterile conditions? Without any microbial infections. If it is microbial infection, it will be completely, tissue culture process will be destroyed, right? Not only this plant, every more than 100,000 cultures can be destroyed if it is any one microbial infection. That's therefore very sensitive process. The aseptic condition very important. Aseptic condition very important without microbial infection. Okay, that's a process. First thing, you have to select explant. As explant, think api gamu banana tissue culture is popular. Right? Okay, we can get as explant the middle of stem plant. Api ඇතුලේ මේවා ගලවලා stem middle part we can use as a explant. Okay, after that it should come from a young and actively growing part. The tissue should be easy to sterile. Yeah, the tissue after is separated this explant from the plant, this sterilization is very important because the surface of explant it can have some infection, microbial attraction, right? All these microorganisms need to be destroyed. If it is any one microbial enter to our culturing process, entire production will be destroyed, wasted. More than millions of money will be, it's a very expensive process, right? Expensive process. Hari, hondata ahaga. Okay. Now think I have separated explant. Apigamu, if I got leaf part, okay, plant leaf part, I separated as explant, and now I have sterile that. Sterile me, I have washed that by ethanol and chlorine solution, and finally distilled water. Then the surface sterilization of explant is done. That we call as surface sterilization of explant, right? Okay, that one is done. Hari. Balum දැන් මොකද කරන්න කියලා. Okay, then after preparation of explant, we have to prepare the nutrition media. Yeah, the nutrition media, it's contained, as I explained earlier, it's an artificial media, artificial semi-solid media, including growth regulators, all nutritions, everything required media is developed. After that, the media also need to be sterile. අපි ඒකත් microorganism ලා අයින් කරලා ගන්න ඕනේ. If it is any microorganism attack to this media, that one also coming problem. Then what we doing, this media autoclave එක දානවා. ඒ කියන්නේ oven එකේ දානවා and heated. Then what happen in the heating process, this microorganism will be break down. හරි. ඒකත් හරි. This MS medium Gambus B5 media are artificially market available media we can use for tissue culture, right? Hari, මේක තියෙනවා nutrition, vitamin, carbon source, everything for plant root development, and it's contain plant regulators, auxin and cytokinin as well. For callus formation, auxin, callus I will explain now. Shoot formation, cytokinin. Shoot formation, cytokinin. Okay, now we have the explant is ready, growing media is ready, now what we doing, now media is sterilized, explant is sterilized, but the instruments make it use can instrument, gloves, everything need to be sterilized before usage. What we doing, all metal particles, metal evapi autoclave, and glasses wrapped by papers and heating, for destroy microorganism, right? Aseptic condition, very important, right? After that, insert this explant into the growing media in aseptic conditions inside the laminar flow cabinet. Okay, I'll show you all steps now. I'll make a practical pen. Okay, after introduce this explant into artificial media, what we doing, it's completely sealed and placed in a favorable condition. Favorable temperature, relative humidity, everything favorable condition we have to place. It's completely controlled condition, artificially controlled condition, right? Artificially controlled condition. Now normally we doing this process inside a beaker or jam bottle like that. Okay, think this is the culture media and this is the explant. This explant. Okay, when it is within 1 week or 10 days, and it due to this culture media, what happened? It's producing new cell. This plant tissue producing new cell, and it's converted into cell structure like this. That we called as callus. Cell structure we called as callus. That is a callus. That's a callus. Okay, then what we doing? Again, get this callus out. මෙන්න දැන් කැලස් එක අපි එළියට අරන් තියෙන්නේ. මෙහෙම සෙල් ගුලියක් වගේ එකක් තියෙන මේ වගේ. Then what we doing? This callus cut into several parts. මේක අපි මෙහෙම cut කරනවා. Small parts. Again this callus growing in different culture medias now. එතකොට අපිට එකම same mother plants, we can same explant, we can get several plants, large number of plants from one mother plant. Body plant leaf part එකකින්. I got. Then again this callus formation and again we can subculture. හරිද මේක normally recommendation is 7 times subculture කරන්න පුළුවන්. Subculturing mean callus cut and place in another medium. එතකොට අපිට ගොඩාක් එකම explant එකෙන් plants 1000ක් විතර හදා ගන්න පුළුවන්. More than 1000 plants can be planted by small explant. Understood? Okay, now what we doing? We change the composition of the nutrition media. Nutrition media composition mean increase the cytokinin concentration. When increase cytokinin concentration, increase root formation. Then again increase auxin concentration, then increase shoot formation. Inside the bottle, this callus form as entire plant. ඒ කියන්නේ අපි banana leaf එකක් ගත්තා නම් ඒකෙන් හැදෙනවා banana small plant of banana, right? After that, but the problem is this okay, that's a process. Make one name, I will explain. I will show you the entire process. Okay, I'll show you entire process. But the problem is this artificial condition produced plants are well soft. Therefore, we cannot directly introduce to the field, outside environment, outside climate conditions. Therefore, the plant should have a hardening process. Hardening process means now the plant is developed inside a jam bottle. Very soft plant is developed. Right, this is a plant nutrition media. Now very small plant is generated with leaf and buds. It's a very healthy plant, but the problem is it is very soft. Therefore, what we doing? We keep shade houses and grow these plants. මෙන්න මේ shade house එකද අපි plant කරනවා. එතකොට shade house mean they can still control condition but not like laboratory. It's little bit sunlight now. Now day to day what happened? It's day to day introduced to normal sunlight and normal conditions. Okay, after 10 to 15 days what happened? This soft plant mature and hard and adapted to normal environment. After that we can feel in the normal environment. I got. It's a very simple story. ඔයාලගෙන් ඉච්චර අහන්නේ නැහැ. ඔයාලගෙන් MCQ එකක් අහුවොත් තමයි මේකේ. Right? This is the tissue culture. Okay, I'll show you now. Then you can easily understand. This is banana tissue culture.
In this video, we will explore the tissue culture process of banana plants, starting from sword suckers and moving through several stages of micropropagation. By the end, you'll understand how to produce
tissue culture plants
ready for cultivation. Let's begin. Extracting the shoot apical meristem. We start
okay,
with
this is the case of banana plant. The first step is explant preparation. බලන්න කොහොමද කරන්නේ?
From these suckers,
okay, this is explant now. But making separate the
this
middle
explant
part.
has two main parts. The lower portion, known as the rhizome. The upper portion, known as the pseudostem. By carefully extracting the inner portion of the rhizome and pseudostem, we obtain the inner shoot meristem, which will be used for micropropagation. Preparing the explant. Next, we place the explant in a glass
Okay,
jar.
this is sterilization of explant.
The lower portion turns brown due to the
Sterilization of
polyphenol.
explant.
To reduce these polyphenol, we treat the explant with a 1% ascorbic acid solution for 2 to 3 minutes. Afterward, we rinse it with sterile RO water 3 times. Sterilizing the explant. To prevent contamination, we follow several sterilization steps. First, treat the explant with between 25% solution for 2 to 3 minutes.
Ethanol, right?
20 is a detergent that helps clean the explant. Wash the explant again 3 times with sterile water to remove any remaining detergent. Next, treat the explant with 70% ethanol for 2 seconds. Rinse again with sterile water to remove.
RO water means sterile water.
Finally, use a 0.1% mercury chloride solution to further sterilize the explant for 1 minute. Again, wash the explant 3 times with sterile water to ensure no residue remains. Nutrient media for micropropagation. In tissue culture, the nutrient media plays a crucial role in plant growth.
Okay, that's a nutrition media.
There are 3 main types of media used in micropropagation. Initiation media, used to establish the initial growth. Multiplication media, promotes the multiplication of shoots. Rooting media, encourages root development. These nutrient media contain both organic and inorganic salts. Macronutrients like magnesium, potassium, and calcium are added in gram quantities, while micronutrients such as molybdenum and zinc are used in milligram amounts. Additionally, vitamins and other organic additives are included to support plant development. Sterilization of tools and excellent. During the process, we sterilize both the plates and
okay, that is a laminar flow cabinet. That's a laminar flow cabinet. And we use Bunsen burner for surface sterilization. Microorganism we using the heat of Bunsen burner, again that we call as thermal sterilization. Thermal sterilization also doing for avoid contamination.
Environment. Once sterilized.
Okay, that's the laminar flow cabinet. See, again this explant cut down. Very small part separate for propagation.
Remove outer coverings and inoculate.
This is explant. Very small plant.
The jar is immediately sealed and labeled with the inoculation date for future reference. Stages of micropropagation. The micropropagation process consists of 4 media inoculated into the initiation media. The jar is immediately sealed and labeled with the inoculation date for future reference. Stages of micropropagation. The micropropagation process consists and inoculated into the initiation media. The jar is immediately sealed and labeled with the inoculation date for future reference. Stages of micropropagation.
Okay. See, they are the stages. The first one, this is a media. This is a culturing. Culturing. Initiation means nowadays develop at cell callus. Now see, it's multiplication stage. Now this is a rooting stage.
The propagation process consists of 4 main stages.
Okay, they are the
Stage 1, collection and inoculation. The shoot apical meristem is inoculated into the nutrient media. This is the initiation stage, where the culture starts its growth journey. Stage 2, multiplication.
Okay, this is multiplication.
After 15 to 20 days, the shoot tips are transferred to multiplication media, where they proliferate and multiply due to the presence of growth hormones like auxins and cytokinins. Typically, benzoylaminopurine is used for shoot proliferation. Over the course of 3 to 4 subcultures, you can achieve rapid multiplication, eventually producing hundreds of new shoots. Stage 3, root proliferation. Once the shoots are about 5 centimeters tall, they are transferred to rooting media to develop roots.
Now rooting media for root development.
4 to 30 days, these plants are ready for further subcultures or transplantation. Stage 4, hardening. The hardened plants are prepared in 2 phases. Primary hardening.
Now plants are ready to plant, but they are very soft.
And kept in polytunnels.
Okay, that's why we use shade houses for control condition.
After 20 days,
We can plant in the field.
5% FYM, farmyard manure, 25%
Now they tissue culture plants.
50% soil.
මේ
These
ඔක්කොම
plants are kept in
මේ ඔක්කොම හැදුවේ මොකකින්ද? Very small shoot part, right? Very small shoot part.
tunnels for further growth, ready for the field. After the hardening process, the banana plants are ready for planting in the field. These micropropagated plants are disease-free.
Now you can understand it's a well-maintained banana plantation, right? What are the advantage of tissue culture? Now can you look in this field, you can understand. Yeah, the main advantage is they are haploid plants. What are the meaning of haploid plants? It coming only mother plant characteristics. Then අපි seed එකක් ගත්තාම it's a sexual propagation. That means it's not mother plant, only mother plant characteristic. It has both characteristics. But in micropropagation, in tissue culture, it's completely mother plant characteristics. If you got good characteristic plants, these all plants have the same characteristic. That's the main one. No any variations. That's the main one. Another thing, we can maintain the uniform growing characteristics. Uniform growing characteristic. Their heights, their growing pattern, everything same. Therefore, very easy to maintain these fields. And they are very no diseases. Pest and diseases free plants we can get by tissue culture. And we can preparation clones. Clone means similar characteristic, large population with similar characteristics, right? They are the advantage of tissue culture.
And available year-round.
See,
ensuring
same height,
consistent yield,
same field, same harvest.
Tissue culture
See?
plants can significantly reduce the risk of plant diseases caused by microorganisms.
Okay. Give me a second.
Tissue culture. In plant breeding, the technique of tissue culture has emerged as a very in this method of autonomy. Each cell is
Welcome to the world of tissue culture, a fascinating field of biotechnology that has changed the way we grow plants. Tissue culture is a technique that cultivates plant cells in a controlled lab environment. It allows us to grow multiple plants from a small singular piece of plants, expediting plant production process compared to traditional methods of seed propagation. More happening for the field is micropropagation, since the same concepts are used in asexual propagation methods. The process begins with selecting a small selection of plant tissue, such as leaf or stem, and sterilizing it to eliminate any microorganisms. This sterile tissue then placed on a nutrient-rich medium containing essential nutrients for growth. Demonstrated shortly. The nutrient medium provides all the necessary elements for the growth and development of plant cells. The most common nutrient medium is MS and includes essential nutrients. We also add sugar, agar, and growth hormones such as pH, which is cytogen and helps cells multiply. Over time, the plant tissue starts to grow and multiply, forming many different growth shares that can cell culture. After the multiplication stage comes where an oxygen such as NA is using this fruiting. After a few weeks,
environment here are of Thai constellations that are filtered in case any sort of pathogens enter the laminar flow hood during their work. Hopefully they are cleaned out before they enter the tissue culture jar.
Welcome to Bioscholar. Today, we'll
Okay. Okay, this is a tissue culture. I try to show you this laminar flow cabinet. This is a laminar flow cabinet. This is a laminar flow cabinet. Yeah, like this is the one. Wait. This is a laminar flow cabinet. It's completely aseptic surrounding, right? Then we can introduce plant part without any microbial attraction involvement, right? It's completely, yeah, this is the shape of laminar flow cabinet. Yeah, like this. Okay, this is the laminar flow cabinet. That's how we're doing. Okay. Okay, the tissue culture is completed. Okay, it is given me application of tissue culture, production of disease-free plants. Yes, it's producing disease-free plants, and clone preparation. Clone mean multiple, large number of plants in same characteristic from same parent, single parents, that we call as haploid plants. Haploids plants. අපි සාමාන්‍යයෙන් seeds වලින් prepared කරාම අපි කියන්නේ diploid diploid plants. Haploid plants we can prepare. Haploid plants. Haploids. අපි seeds වලින් ගත්තාම ඒක diploid plants. Diploid plants mean both plants characteristic same. Mother and father. Parents දෙන්නක්. දමු ඊට single parents. අපි එක plant එකකින් part එකක් ගන්න. Then all population same characteristics. That's the main advantage. Then the maintenance, everything's very simple. The harvest also same. All characteristics same. Nutrition requirement, everything same. And conservation of this may are වන්ද වීගන යන කියන්නේ ඒ වගේ plants, rare plants conservations, this method very important. The production of secondary metabolites, like alkaloid and flavonoid. Yes, genetic modification and development and genetical modification also, this tissue culture we can use. Mainly remember these two characteristics to production of disease-free plants and clone preparation and conservation of endangered plant species. Okay, that's the tissue culture. The advantage, no, what are the disadvantage? The main disadvantage is if it is any microbial infection, what happen, complete population will be destroyed. That's the one disadvantage. The other disadvantage, it's highly expensive process. අපි දැන් seeds ටිකක් grow කරනවට වඩා it's highly expensive. That's the other disadvantage. The other thing, you should have full complete technical knowledge. Without the normal farmers can't do this techniques, right? Okay, they are the real time disadvantage. All right, let's move into final parts. Okay, vertebrates and invertebrates. That's a very small part. Now I have a recording of 1 hour recording of this part. I will send it. This part very simple. Vertebrates and invertebrates. That means birds, mammals, and fish, and their economical values. Okay, what's the recording of vertebrates and invertebrates? Then this lesson is completed. Now, yesterday and today we did more than 7 hours. More than 10 hours we did. Okay. Okay, I'll send the vertebrates and invertebrates recording. Then the cell structure is completed. Next day, next Friday, we start our final lesson. It is only 4-hour lesson. Then the theory part will be completed. The next final lesson is environment chemistry. Environment chemistry, it is a simple lesson. අපි දැන් කරපු ගොඩාක් දේවල් අපි කරලා තියෙන්නේ ඒකේ. More than 50% actually we have completed. Nitrogen cycle again, wastewater treatment again is there. අපිට තියෙන්නේ acid rains, photochemical smoke gas, acid rain, photochemical smoke gas, global warming. That's all. And ozone layer depletion. Ozone layer depletion. ඒ ටික කරලා ඉවර කරන්න තියෙනවා. Now syllabus almost completed. Syllabus almost completed. Let's meet final part of final lesson. Now today we can't start, very tired now. Let's do final lesson on this Friday. This Friday we can complete. This Friday ඉවර කරලා දාමු. Okay. Please go to this small recording, vertebrates and invertebrates, mammals and arthropoda economical values. The small recording, go through and complete cell structure. Make sure to prepare a short note. Amina, make sure to prepare a short note of this entire lesson. Now, the plants parts of the diagrams and the story of secondary growth, you already can understand by me. You can already understand by these diagrams. Okay, then let's meet Friday with our final lesson, environment chemistry. Good night.